So thank you so much, participants, for registering for this webinar, and thank you, Ganesh, again for accepting our invitation. Genomics, as all of you know, that is a startup which we have started with the aim of sculpturing your research. We have been recently incubated at NSR Cell at IIM Bangalore. We are providing bioinformatics services and developing a lot of solutions for your research. If you have any suggestions, ideas, requirements, and if you want to know more about us, you can Google us, or you can write back to us, or you can join us on any social media page. We are quite active on LinkedIn, Instagram, and Facebook. So you are most welcome to give us a call right away and talk to us about our ideas. And as I've said, we are aiming at providing bioinformatics services and developing solutions for life science researchers and the healthcare industries. There are many upcoming ideas, new ideas, and we are developing a lot of solutions. We are always open to ideas and always open. So we are looking for bright people to join our team as well. The major key competencies that we provide are the data analysis support that we provide is bioinformatics support. We are having expertise in next generation sequencing analysis, and we do provide a lot of training solutions also on such topics, including a lot of workshops and webinars that we regularly conduct, including hands-on training programs, long and short-term trainings and programs, and also dissertation and thesis projects. So if you are looking for a specific topic that we have not yet covered, we are always open to suggestions, and we'll be happy to conduct such trainings for you and your college group or your friends. Just let us know what you are looking for. As mentioned, we are also conducting a lot of professional courses as well. I've mentioned a lot of them in my previous slide. So the other ones that we do conduct are clinical research and structural bioinformatics. We recently conducted two very nice and successful workshops on structural bioinformatics and basic bioinformatics as well. We are soon going to conduct a small interactive training program on NGS analysis. If you have any requirements, do let us know. So before moving forward, telling you much about genomics, let's take out some time to introduce our speaker for the day, Dr. Ganesh. Thank you, Ganesh, for joining us. Ganesh is a computational biologist and bioinformatician in the area of plants and animals. His major areas of focus are genomics, epigenomics, and regulomics. He's currently working as a postdoctoral scientist at Division of Biology, Kansas State University, Manhattan. He has done his PhD at CSI Institute of Himalayan Bioresource Technology, which is also known as IHBT in Palampur, Himachal Pradesh. And he has done his MSc in bioinformatics from Sri Shivaji Science College and BSc from the same college. And he has a lot of awards and fellowship, and he has been a first rank holder in his MSc. And he is also was an Inspire Fellow of Department of Science and from Department of Science of Technology, Government of India. So thank you, Ganesh, for accepting our invitation. And we are looking forward to your presentation and your talk on the tiny molecules that derive gene regulation. So Ganesh is going to discuss about the part of the DNA which was previously known as junk. So everybody thought that this is huge portion of our genome which is known as junk. But later on, people identified a lot of small RNAs that are actually functional and have a lot of functional roles in various. They have a lot of functions. So he's going to talk about those small RNAs in both humans and plants, and he's going to discuss about the biogenesis, that how they are going to get generated, and also he's going to throw some light on the computational analysis of the known and novel microRNAs, how to identify these microRNAs, how to predict the targets, and how to, if we have these mRNAs and their targets known, what are how do we identify the pathways, how to do the pathway enrichment analysis. So Ganesh, thank you so much again. And now I'm going to make you the presenter so you can share your slides. Also, participants, if you want to ask any question, you can write that down in the Q&A box, and also you can raise your virtual hands so that we can unmute you and you can ask the question. Over to you, Ganesh.
Okay, thank you. Let me share my slides.
Yeah.
Can you see now?
Not yet. Yes, now I can see.
Okay. So it's in the full screen mode?
Yes, now I'm on full screen.
Okay. Thank you, Diksha, like inviting me for this talk, and also thank you for all participants who are interested to listen microRNAs. So I'm Ganesh Pandade, working as a postdoctoral scientist in Division of Biology, Kansas State University, Manhattan. So basically, I'm working on the microRNAs in from my PhD tenure to still in our lab. Now we are working on the C. elegans model to identify the microbiogenesis, different types of microbiogenesis biology in our lab, like working or like developing different types of strain. So I do data analysis, mostly NGS data analysis. All the time I'm working on the microRNA, small RNA sequencing data. So let me start my presentation. So basically, what type of different teeny molecules or small molecules in cell system? The first name comes, that is microRNA. Those are like near about 21 nucleotides. Then second, small interfering RNAs, mostly found in the plant system. Then RNAs, and the transfer RNA derived small RNAs. These all classes identified these days, and lots of researchers working on all types of RNA. So we are going to talk about the microRNAs in next slide. So microRNAs are small in the length. They are like around 21 nucleotides, double-stranded, and present in hairpin loops RNA structure. Also, there is 3' 2 nucleotide overhang present, like they um, formed due to the DROSA and dicer cut. And also there is a seed region, which is most important part of all this microRNA, because that seed region of 7 nucleotide form the complementary bond with the mRNA and just stop the gene regulation. So these microRNA also, like they are tissue specific, condition specific because they taught to switch on, switch off the gene regulation according to cell signals or cell requirement. And this microRNA presents in conserved or poorly conserved families, like they are conserved throughout the evolutionary course in different animals. So all these characters are really interesting, and we can see those microRNAs really important in the system. So the first microRNA identified is LIN4 in Victor Ambros lab from C. elegans. From that onward, the microRNA got like lots of attention throughout the different labs, and now we can see the MIR-base almost 25 to 3,000 microRNAs in human identified, like through the validate techniques or prediction methods. So how this microRNA generated? So microRNA generated through the different types of biogenesis pathway in animals, plants, and C. elegans. So if you see here in animals, initially the primary transcript of microRNA generate transcribed from the DNA by RNA polymerase second, and then this primary microRNA cleaved by the DROSA. Also, there is help from the PASHA enzymes, and then again, this primary microRNA cleaved into precursor microRNA cleaved and exported into the cytoplasm by the X protein 5 and dicer, and after dicer cuts, it formed microRNA mRNA double-stranded structure, 3' 2 nucleotide overhang. The red color shows the guide microRNA strand, and the black shows the microRNA star strand. They depend on their signal which strand is going to load into Argonaute protein, which is basically a complex protein that binds with the microRNA, or they form a complementary bond with the microRNA bases, and that complex is called like Argonaut Rix complex, RNA inducing silencing complex, which is basically bind with the mRNA in 3' UTR, specifically in animals, and just stop the gene regulation by transcriptional gene silencing or post-transcriptional gene silencing, or either the microRNA just degraded. But in plants, this microRNA is generated through like little different pathway. Like the main difference in the DROSA and dicer enzymes. Here, DCL1 dicer 1 act on the primary microRNA and generate precursor, and then again dicer just cleaved and form the microRNA mRNA duplex structure, and then this microRNA structure export into the cytoplasm and does all these things in the plant as well. So the difference RNA binding factors, the initiation enzymes, they are responsible throughout this biogenesis, and they really act as a driving like we can say the main factors that microRNA regulate the gene regulation using these factors. So the same biogenesis in the C. elegans Reinhardti or C. elegans, the same is that the main change is the different types of enzymes. They change according to animal. So they are the basically same types of enzyme, but that they change some kind of like protein structure and important in microRNA biogenesis. So these are all the steps that microRNA generated. The main pathway, or this pathway, is basically called canonical biogenesis pathway. There are some microRNA they are also generated through the non-canonical biogenesis, like they bypass some steps from this micro biogenesis, and still they show some kind of like same kind of the properties of microRNA, and they do all these functions, same functions in the cell. So what type of structure does microRNA have? So microRNA is basically generated from the DNA in the form of primary transcript, and then it cleaved into precursor microRNA, which is basically around 70 nucleotides or 70 to 80 nucleotides. Some of them like more than 100 base pair as well in present in Mir base. And then this precursor microRNA is cleaved, processed by the dicer enzyme into 21 nucleotide, 21, 22, these all different lengths. So if you see this structure is present in stem loop, the loop structure, and there is a big loop structure. Again, it's just fold to itself and form a duplex strand. So DROSA act on the 5' side as well as 3' side. So the upper arm is a 5p and 3 the folded strand below strand is 3p. So basically it's in duplex form, and this this DROSA enzymes cleaved here and here, all from above and below sides, and just form the duplex structure. So the computational representation for this structure is like this one, in the like dot bracket form. The dot represent the loop and the bracket represent the complementary between the above and below strand. So this is like stem loop structure of microRNA. Basically just shows the stability, thermodynamic stability of microRNA. So we said that in the initial slide that microRNAs are conserved because they show same kind of or similar identical sequence similarity within all this microRNA member of a single family. This is the LEAD7 family, and this one is the seed region. You can see at the below, that is conserved seed region from 2 to 8 nucleotide, and this present in all this microRNA. basically they conserved during the course of evolutionary process. If you want to find the same microRNA with same seed region also present in different animals like C. elegans, in mice, even in the plant as well. So different types of family, they are conserved, some of them like strongly or poorly conserved families. There are some other families that are non-conserved because they don't show the same seed sequence, but some kind of partial conservation in their sequence. So lots of family, more than like 100 families of microRNA available in current annotation of MirGeneDB. If you want to browse this, like this section is downloaded from the MirGeneDB 2 database. So what type of sources that provide the microRNA information? The first one is the MirBase, which is basically developed to provide all the microRNA information. Nowadays, this microRNA MirBase database also provide target information, then gene ontology, functional enrichment as well. It includes like most of species data from the NGS as well as validated methods, and provide all the like sequence information and all the things. Then again, the MirGeneDB, now it's out, like MirGeneDB 2 is out and provide all.
Ganesh, sorry to interrupt you in between. You are on a presenter view, so we can see both of your slides, the next slide as well. So can you remove the presenter view?
Okay, sorry.
No problem.
Oh, I have the double screen, so that.
Yeah.
Let me
just change the presenter view. And also we have a question from one of the participants that how do we have to download miRNA data? But you are already covering that. So I think that
Yeah.
will be. Yes.
Oh, sorry.
This is perfect.
Okay. Let me resume. Okay. I will answer the question once I will finish all this thing like presentation, and then we can go one by one. Yeah. Okay. So I'm talking about the microRNA information resources. So MirBase is the master repository for all this information. You can download sequence, then their structure, then target information, even gene ontology, functional enrichment information as well. Even the lots of links also available there to like that microRNA came from the different types, like this study or this study, they provide the references from the PubMed as well. So you will get all this information if you search the entry in the search box. Like if you are interested in LET7, just type HSA LET7. So it provides some kind of annotation technique, like it's initially the microRNA name start with the species name, like 3-letter code prefix. That is, if you are working on the human, there is like HSA. If you are working on the C. elegans, that start with the cell. So these are all the nomenclature available in the MirBase. Then recently the updated version came out of MirGeneDB 2.0, which is pretty interesting and also providing the same kind of information with the expression data as well. MirBase also provide the NGS data like expression from the small RNA sequencing, and as well as MirGeneDB also provide the same thing. If you want to search all articles related to the microRNA, you can go to the eMirIT platform, which is basically a text mining platform to provide information of all these research articles that coming these days or from the last. And then Tools for Me is another repository. Basically, it's a platform to provide the information about the tools and web server related to microRNA. There are different types of selection criteria there. You can search all these platforms on these links on this server. So the main what are the functions of microRNA? So we are talking the microRNA is important and previously not known, but now it's everywhere working on the microRNA. So it's well known and what are the function they do. So microRNA basically target to mRNA in 3' UTR in animal system and just stop the translation process, like it's not going to generate or just produce the proteins. So basically controlling the gene regulation, gene expression through the gene regulation. So cell does have particular types of signals, what type of function they want to do. So they switch on the genes according to that. So the different types of microRNA, like they process, they generate, they target to the mRNA, and just do all these things. Like microRNA also perform the gene regulation, development, growth, proliferation, and it's basically like switching the gene expression according to the cell system. And this microRNA, like some of the microRNA, they're important. If the microRNA's expression goes down, some of the cancer, like in disease conditions, some of the genes upregulated, and they form tumor cells, which is basically a disease condition. And in case of plants, due to the upregulation, downregulation of microRNA, cause the stress condition. So the plant is like in the alter condition, or like we can say the susceptible or the stress condition due to different signals. But in the like here I said that I'm gonna cover that. So the roles, like currently we are like studying the microRNAs and the important in the SARS-CoV-2. One of our participant published the paper on the microRNAs, that they are some kind of a role in SARS-CoV-2. So basically microRNA, like here in this paper, microRNA enhancement or microRNA suppression. If the microRNA is upregulated and the genes responsible to lead towards the SARS-CoV condition, that microRNA, if we induce or into the or just mimic into the cell system, they are gonna stop that cycle, that pathway, and stop the COVID spread or COVID condition, we can say. And if the microRNA, if some of the microRNA, they are responsible, they are targeting to the genes that are like that causing the SARS-CoV, we can just downregulate their expression and just switch on the pathway that produce the protein responsible to work against the viral infection. So lots of microRNA these days, they're identified in SARS-CoV-2. They perform the roles of all this control. We have like more than 100 microRNA I saw in an article. They are really important in this SARS-CoV-2 as well. So the next is how they target to gene in their UTR region. So there are different theories, not different theories, different methods they target into the mRNA. Like in animals, the targeting is like a seven base pair complementary bonding to the mRNA UTR region, either partially or the totally perfect complementary. So you can see here, the below is the microRNA seed region, and the above line is for the mRNA, is form the complementary bond and just stop the gene regulation. But if you see in the plant case, the total microRNA sequence is responsible to target the mRNA. So it's basically full base pairing binding to the mRNA. These are the main difference in the animal and the plant. So these microRNA target to many genes as well as many microRNA target to single gene. So basically it's one to many, many to one relationship to regulate the different types of pathway in the system. So microRNA can like they do co-expressive as function as well, off target function as well. Sorry. So we learn about the microRNA, how they generate, what they do. Now we are going to move towards the data analysis part, like computational analysis of all these microRNA from the small RNA sequencing data. So basically, all the microRNA they identified from the, like we can also say that prediction of microRNAs falls from small RNA sequencing data from different samples or different tissue, like, or whatever the data type like user generates. So basically, this is the workflow. Initially, the small RNA sequencing data comes from the NGS platform. And then these small RNA sequencing libraries assess for quality, and then we perform the filtering. And this filter reads, survived reads map to genome using the reference genome. And then the future extraction, like different microRNA does have different features. Seed region, then the thermodynamic stability, then hairpin, loop structure. These all features considered when to predict the microRNA from the small RNA sequencing data. And then once the microRNA is identified, we do the expression quantification, basically saying the microRNA is upregulated or downregulated in the data. So in next study, we are like discussing data and how the quality assessment perform. Initially, the raw reads comes and we perform the quality check, like quality assessment by FastQC tool. If you are familiar with this tool, you can easily handle like the commands, or this tool also provide the graphical interface. You can just start and then upload your read sequencing data and just check the different parameters there, like GC contains the freight quality score, and the adapter contains or any contamination in the read sequencing data. If you found something, you can like correct those reads using the trimming filtering step. So after the quality check, we perform the filtering, like quality filtering. Basically, what we check the quality score at every base pair. Suppose we have 50 base pair read length, so we check that every base in that 50 base pair read. So suppose this is a 50 base pair read, and we are checking the from start position to end throughout this read sequence, basically identifying the quality score. Like generally we set 25 quality score, and it's covering 70% of its length. Means from the 50 we at least 40% bases they qualify 25% Fred score. So these all the things considered for the quality filtering. And we also clip the adapter sequence according to library preparation protocol. If we generate the sequencing from like own, or like we do outsourcing, they provide the all this information, you can easily do the clipping of adapter. And the quality filtering we do using the CurdAdapt, FastX, and TrimGalore tools. You can do these things in standalone platform as well. So after the quality filtering, we get the clean reads, and then these clean reads selected between 70 to 27 nucleotides, because the microRNA, they do have like median length around 21 to 22 nucleotides. So in this length, they cover all these reads. After the size selection of all these reads, we collapse identical reads to form clusters, and we also just take the count of all these cluster of identical read sequences. So after filtering, the next step is the genome alignment. We know that all the reads, they coming from the genome, they like they are any type. So we initially map to the genome using the Bowtie. Bowtie is specifically used for the to map the read sequences. You can also use the another aligner, but generally the most of the studies, they recommended the Bowtie. So we map the read with 0 or 1 mismatch. If you are like identifying the novel or known microRNA, you just go with the 0 mismatch, because we need the perfect sequence match in the data with the reference sequence. So after mapping the read sequences using this parameter, we also exclude the other non-coding RNAs overlapping read sequences, because they are the part of like any type of RNAs, no RNA, tRNA, rRNA. So we just exclude them, and whatever the reads survived, we use for the microRNA identification in different tools. So in the next slide, I'm talking about like what type of tool to use for the identification of novel and known microRNA. The main two tools are MIRDIP2 and MIRGE3. MIRDIP2 is the second version, came out in 2012, but MIRGE3 is the latest version, which is basically came out in 2020. And it's provide the different types of module to identify all these microRNA from small RNA sequencing data in the local system, a standalone system. So MIRDIP2 basically takes the input read. Also, we need to provide the reference genome, and it performs the identification of prediction of microRNA based upon the read stack. Like it's just from the read stack from the 70 base pair location, and it just search if this is a 70 base pair, and search for the read stack, first read stack and the second. If it found some type of stacking of reads, that it just excise the precursor and characterize the RNA structure, and as well as the RAND value to to predict that this microRNA is really true microRNA or not. And after the finding of this microRNA, just quantify the expression matrix, which is basically used directly in the differential expression packages, or you can also do different types of calculation on this matrix. So this MIRDIP2 is available in 3 different modules. The main one is MIRDIP2, and the one is mapper module, used to align the read sequences, check the input file, align the read sequences to the genome, and the quantifier module directly used for the quantification or expression of microRNA of known and novel from the data set. And the MIRGE3 platform is like advanced platform. It's run like on the command line as well as in graphical. It takes input reads from multiple libraries or multiple data sets. Initially, it's annotated reads. It just separate the annotated reads and unannotated reads, and then find the known and novel microRNA, as well as isomer does identification of isomies, which is basically called as a microRNA variant. And from the unannotated reads, the tool identifies the novel microRNA and then generate the expression matrix and perform the differential expression using the DE6 package 2 as well. So in the next slide, I will show you how to run how to install MIRG3 and how to run this pipeline on your own data set. This is basically developed in Python. And if you are familiar with the Python installation, so you can just type pip install MIRG3 and it just install in like 20-30 seconds. And then you can run this pipeline by providing your input files, like -s option is there to provide multiple small RNA sequencing filter reads file here, like input 1, input 2, and so on, separated by comma. Then MIRG3 like provide the own library according to the reference genome. So if you are working on the human or mice or plant, just go on this link and download all this library. So this library contains different types of models they already generated from the different features of microRNA and use in the prediction. And also use the database information. If you provide option -db and mention their MIRBase, or if you want to work with the MIRGINDB, just mention here. So it's basically just fetch all the known microRNA information from that database. And you need to provide output folder, any type of whatever you are interested, and also provide the GFF annotation of isomies in GFF format, which is basically advanced format to provide information of different features of the transcript, or we can say any type of genomic features. And also provide the novel microRNA information, then AI to I editing. Also provide the let me show. What is that? Oh, the output file in the FASTA format, basically in Collapse. And also if you are less in memory, you can also provide the option CMS. Basically 2000 is just a memory size, RAM memory size. These are all parameter. The MIRGIN3 it does have different parameters. You can just write MIRG3 and help in the terminal. If you are familiar with all these terms, you can easily search the manual MIRG3. So after this run of pipeline, we get some kind of result. Let me show you. I will show you the results, how MIRG3 provides. Can you see my screen?
Yes, yes, we can. But we can see the slide.
Let me pull over here.
Now, yeah.
Okay. Yeah. So the MIRG3 provides the HTML visualization of all the results. In the first tab, they provide a small RNA distribution, like whatever the samples you process, they provide the distribution of all these RNAs. Like if you see here, the bar line is just divided into different RNA annotation. You can see here microRNAs, ncRNA, rRNA, snRNA, and the main that we are working with microRNAs. We generally just focus on the microRNA reads. If that library does not contain maximum reads belong to mature microRNA, maybe it's a possibility of some kind of contamination of rRNA or something. You just need to check it. So these are all the distribution provided in the MIRGIN3 in first tab. Then also calculate the median read length. You can see here the most of read belong to 21 to 22 from all the libraries, most of the libraries. And then also provide the isomer results. Like isomers, they are either are perfect match to their parent sequence, or they some kind of SNPs present in the sequence. So it's also provide a classification of 3p, 5p, and both type of isomer with or without single nucleotide variation in the data set. Also provide the information of top 20 mRNAs isomer information in this heat map. And then what are the abundant microRNAs in all these libraries you give for the identification. So basically this map showing the expression in RPM format, RPM score, and read per million. And then also provide the information of novel microRNA in the table format. You can see what is the probability. This is the microRNA. The probability is 0.98, which is pretty much higher, and it's coming from this part of genome, and this is the sequence, and how many reads belong to this microRNA. If you click here, it's also provide the information of structure and all these parameters predicted from the model. You can see, let me zoom. This is the hairpin loop structure of microRNA, and also a systematic representation of sequence. Like above part is the precursor sequence and their dot matrix structure, and the microRNA is identified from the 5' end and the 3' end. These are the reads and their respective count here. So these are all the results generated from the MIRGE-3 pipeline. Then once you identify the microRNA, what you going to do? Basically, if you interested in the function and analysis, because we want to know that what are these microRNAs doing. So we do the target analysis using the different tools. Either they are the web servers or the standalone tool. So the first one is the target scan. We basically use the seed region of microRNA to identify the targets from the transcriptomic data for human, mice, or the worm, or Arabidopsis, or any type of organism. So target scan available in different variant. Like it's also provide analysis for mouse, worm, fly, fish, and it's also provide a standalone script to run on your own data set by providing the input microRNA file and the 3' UTR region. So this is basically the most popular platform to identify the target in animals. And there is another one, psiRNA target, basically used to identify the microRNA gene interaction in the plants. So it provides it.
The presenter view. Can change the view.
Oh, sorry.
Yeah, great.
Yeah. So psiRNA target provide the flexibility. You can also input your microRNA sequences and the target sequences, and it will give you results of all the possible interaction from their server. So basically you just download all these results and do whatever you want to, like, with the results. Like you want to form the microRNA gene co-expression network, you can also do that. And if you want to perform the function and analysis, you can use the target gene and put into the WebG stalled server, which is basically used for the gene enrichment. Either it's a gene ontology or pathway enrichment. It also shows the disease information, all this information. This web server is user friendly if someone is not familiar with the bioinformatics part. They can use this web server, or if someone like familiar with the R scripting, they provide a WebG stalled R server. R package is basically run in the R, and you just need to provide your gene list and select your database and just enter the in the terminal. You will get all the results in quick time. So you can also browse with the different types art, but this one is the user friendly. So I use the R version of this tool. So it gives me the result within quick second. Okay, that's all. So this is that one is the last slide. So thanks for listening me, and I'm ready. I would like to take questions now.
Thank you, Dr. Ganesh, for such an, I should say, inquisitive and such a nice, insightful webinar. And you have covered all this information about starting from biogenesis to everything of microRNAs, and you have covered the computational analysis in such a short and crisp way that all the students or the participants who are not even a bioinformatician, who is not a bioinformatician, will understand that how do they can identify these things. So we had a question from a participant that how to differentiate between mRNA-seq and mRNA-seq data, and you have already covered it. Maybe you can just elaborate it in a better way.
Let me check the question. Can we present how do we have to download mRNA data? How to differentiate mRNA-seq from mRNA-seq data? Okay, I'm going with the presentation. How do we have to download microRNA from the Mirbase? Okay, can you see my screen?
Not yet.
Now?
No. You have to share your screen. You have not shared your screen.
Yeah, I'm sharing. Can you see now?
Yes, now we can see.
So if you are on this is a Mirbase database, and it provides all the information here, like search information as well as browse. Whatever the species you are interested, you can go directly to the browse option and just expand all these things. And if you are working on the like Arabidopsis, just click there. So basically it provide all the microRNA information in the tabular format. And if you want to download the sequence, like precursor sequences, this is like stem loop sequences means the precursor. And if you want to download mature sequences, just click on the mature and like select all phase sequences. You'll get all the sequence information from here. So it depends upon the what type of species you want. Just do like this, and you will get all the sequence information. Then if you want to interested into particular microRNA, then you can click on that entry and browse all those things like RNA structure. If you want to relate with the other RNA information, like from the RNA Central or RFAM, you can just click on those links. And if there is a target information available for particular microRNA, they also provide the link, the target information for the listed microRNA, and you will get directly all the targets from that link as well. So this is the way to download the sequence. And the other question, how do we differentiate mRNA-seq from mRNA sequencing? So basically, mRNA is the different one. So if you want to predict the microRNA from the mRNA sequencing, you can predict the precursor sequences because precursor is long, and the RNA, like mRNA sequencing reads also the the longer ones, and you just map to the genome and check the identifier like overlapping sequence with the precursor microRNA. So, and then you also need to consider the all the structure information as well, and the other feature of microRNA. So mRNA sequence is little bit like nowadays old to identify microRNA, but generally most of the like the NGS variant is the small RNA sequencing. So people do this small RNA sequencing and identify the microRNA sequence. But before the decade ago, they used the mRNA-seq to predict the precursor sequence. And you also need the microRNA sequencing, small RNA sequencing, because if you predict the structure, you also want to know that that precursor is responsible for the microRNA generation. So you basically check the microRNA read accumulation from the same precursor in the sequencing data. So this is the way to separate all this, differentiate the things.
Also, there was another way that we used to use previously, that if you want to see, you can also check the coding potential of a particular sequence that you have identified, and that if the coding potential is below the specific threshold, you can specify that that particular sequence is a coding coding RNA or non-coding RNA, and then you can check the length of, because mRNAs are precursor microRNAs of a certain length size, so you can always do that as well. But as Ganesh has also mentioned, there are several other ways to do that. So there is
the features, the microRNA features are important because there is possibility of 70 nucleotides the RNAs, like they are degradation product as well, or random RNA sequences.
Exactly, because the length, so many of the precursor microRNAs might have the same length as the smaller long non-coding RNAs. The long non-coding RNAs, which are the smaller, which are like almost 200 base pairs, might also be in the same length. So now let's take the next question by the same participant. So in the NGS data, how much cutoff value we have to give if we are doing from the cDNA? And so
you mean this question is related to the sequencing, like generating the sequence library or something? Or you want to just like set the criteria, like if there is a sequence, small RNA read sequence there, we need to select, or we need to fix some kind of like read count criteria, like we do in the differential expression. If that microRNA is there, what do? We scan the the read number from the samples, and if we want like the microRNA that belongs or shows 5 reads, at least 5 reads or more than that 5 reads, we consider for the expression analysis. So I don't know, like if we are doing from the cDNA. So clarify if you want to know the library or library preparation or the NGS things to filter out the microRNAs.
I unmuted you, Lakshmana. If you want to speak, you can directly speak to the speaker. You can unmute yourself and ask your question.
Okay, ma'am. Actually, so when we are isolating RNA, so we will isolate we will isolate from the cDNA, means cells, right? So that time, so how we can differentiate whether we are isolating mRNA or total cellular RNA?
Like, so there are some sequencing kits. I think they have like protocols for the separation of all these mRNA, microRNA sequences from the DNA. So they depend on the sequencing kit. So if you like go through that manual, you will get all this idea.
Okay,
I guess.
Okay, that's fine.
This part is about to generate the sequencing libraries.
Yeah. Atul, can you take this question?
Yeah. So basically, what happens? The time you are basically most of the kits help you to isolate the total RNA. Okay, and if you use the triazole method, most of the time you'll be getting the total RNA. There are kits from Kaizen and a couple of other kits also, and mainly they mention that why this kit is being used. So mostly, as Dr. Ganesh said, you have to go through the manuals, but mostly you will get the total RNA, and then you will do the cDNA prep. And when you do the cDNA prep, that will be targeted according to your particular target region, and it will be not towards any other region. So you can target there the microRNAs or whatever region you want to target. But initially, you will go through the total RNA, then you can target the microRNA part, and then you can so on do your experiments.
Even in microRNA also, we have to convert into cDNA?
Yes, you have to.
Okay, that's fine.
Thank you, Atul, for this answer. So we have another question from next participant. How to predict the role of a particular microRNA?
As I explained in the slide, the role of like targeting analysis. If you able to find the targets of microRNA, and the microRNA target basically they are the genes. So if you identify the gene ontology or pathway enrichment of all these target genes according to their differential expression, like some of the microRNA, they are upregulated and their targets generally downregulated in the system according to expression data. So you can predict that these microRNA are upregulated and they're targeting to the gene 1, gene 2, gene 3, and they are downregulated and responsible into pathway 1, pathway 2, pathway 3, like that. So you can predict that role first to identify the targets and then do the functional enrichment through the tools or the available web server that I explained, like Target Scan. If you are working on the human animal models, you can use Target Scan. And if you are working on the plants, you just use the PSI-RNA target tool. And then the web package, and as well as the web server or WebGStall, which is basically identify the role of microRNA, means the gene enrichment and the pathway enrichment. And the next one, MIRDIP to identify the canonical or non-canonical. These things are like they are not differentiate like how they generate canonical or non-canonical microRNA, because the tool is know to predict the whatever they take input. So we have to we just want to know that this microRNA, predictive microRNA, is canonical or non-canonical. We need to go through the wet lab techniques. Either this microRNA is generating through the DROSA cuts, dicer cuts. Then we can know that, or we can separate all these things. But the tools, it just provide you the prediction of all sequences. Is that sufficient for you?
I hope that answers the question. So if is there any other question?
No.
I don't think so. So thank you, Ganesh, for this insightful lecture, and thank you for your patience to answer all the questions as well. So I've also started a small poll over here. So participants, please rate the lecture, and if you are interested in a detailed webinar or workshop or a course on this in any field related to small non-coding RNAs or any other related field, feel free to reach out to us, and we can also ask Ganesh to take a few lectures of the course as well. So if he has time, whenever he has time, we can arrange that course. So please feel free to reach out to us if you have any more suggestions or if you have any such requirements. And if you have, we'll be sending your certificates along with the recordings of today's webinar within 1 week. And do write to us if you want to connect to us or if you have any suggestions. Thank you so much.
I just like to mention something. We are also providing the service of NGS data analysis. So if someone interested to do outsourcing for the analysis, they are most